Hello, my name is Katherine Schneider, and I am a first-year PhD student from the College of Forest Resources and Environmental Science. My research concerns genetic applications in entomology, and revealing silphid stomach contents using novel iDNA methods is an important preliminary work for the rest of my project. A major focus of this project is using invertebrate DNA, or iDNA methods, which is DNA obtained from invertebrate stomach contents and is used frequently for vertebrate biodiversity studies because of the less invasive nature of iDNA techniques over traditional trapping methods for other animals. Additionally, iDNA methods usually are less time-consuming, and iDNA studies have been conducted with carrion flies, ticks, and mosquitoes, and our work is a novel method proving the viability of silphid beetles. Silphids of the family Silphidae in the order of Coleoptera, or beetles, with the two major groups being burying beetles and carrion beetles, were chosen due to their diet preferences. Burying beetles are more often found on small carcasses, such as rodents or birds, because they use the carcass as a sort of nest underground in which they raise their young and feed on the carcass. Carrion beetles are found on carcasses of any size and don't exhibit preferences. In our trapping, we set up pitfall traps in 18 plots, with 2 traps per plot in the Northern Hardwood Silviculture Experiment for Enhancing Diversity, otherwise known as NH-Seed, which is a part of the Ford Forest in Alberta, Michigan, with different logging treatments as shown in the map at the bottom of my poster. We obtained around 40 beetles from trapping in the summer of 2019, using traps that follow the model as shown at the bottom of my poster to the left of the map, which include a mesh screen between the bait and the captured beetles to prevent DNA contamination from the raw meat bait that we used to attract them. Once we had the beetles, they were swabbed in the field and their stomach contents were dissected in the lab to be analyzed with DNA extractions, followed by a polymerase chain reaction, or PCR, for DNA amplification using 2 different universal vertebrate primers meant to amplify only vertebrate DNA. Visualizing the PCR products on agarose gel using gel electrophoresis shows that we have successfully extracted vertebrate DNA, but Sanger sequencing still needs to be done to determine the identity of the vertebrate species. Once the sequences have been processed, we will be able to compare our results to small mammal trapping data that also was conducted at our study sites, and we will be able to complete the publication for this work to submit to a wildlife or entomology peer-reviewed journal. After this work has been completed, we will be expanding sampling of silphids in addition to sampling for ground beetles in the upcoming 2021 field season, and we will use high-throughput metabarcoding sequencing on Illumina-MiSeq platform for genetic analysis instead of Sanger sequencing. This concludes my presentation. Thank you.